In 1953, scientists discovered the structure of DNA and changed forever our understanding of genetics, of heredity, and even of life itself. It was the beginning of a revolution in biology that led eventually to the sequencing of the human genome, the genetic code of life that defines our species. But this was a revolution with a difference. These critical scientific discoveries were among the first to be documented on television and radio.
Dr. Wilkins, what do you actually do?
The main thing in my kind of scientific work is to be able to fiddle with a thing, go on fiddling with it, and fiddle until everyone else is given up.
In an era before spin doctors and PR, the scientists were prepared to talk about their work and each other with extraordinary candor.
She came toward me, I thought she was going to hit me.
Oh God, who hit who?
Their stories, buried in the BBC archives, reveal the people behind the science.
In a few moments, through television, you will be able to meet them and judge for yourselves what manner of men they are.
Passionate, ambitious men and women, driven by intense, sometimes bitter rivalries.
Scientists sat back and got fat and happy getting tens of millions of dollars, and I put John in that category.
We have already released 2/3 of the human genome into the public domain, and he's released nothing at all.
The archive reveals that truly to understand how the discoveries were made, it's essential first to understand the people who made them. This is the story of those scientists, of the men and women who set out to crack the code of life.
Happy days.
Told in their own words.
Do electrons think? Electrons are charged particles, the minutest we find in analyzing the ultimate constitution of matter. To think that such a particle can think is so absurd that I might give the answer no and have my talk over. However,
this is one of very few recordings made of a man called Erwin Schrödinger.
not of course in this
Schrödinger had served in the Austrian army in the First World War, and later made a name for himself in physics as the inventor of a theoretical cat. The odd thing is that here is Schrödinger, a world-famous physicist, musing on a biological problem, the nature of consciousness, of what it means to be human.
Mind per se cannot play the piano. Mind per se cannot move a finger of a hand. This leaves us with the outlook that our body is as automatic or non-automatic as any inanimate piece of matter, only infinitely more complicated than even the most ingenious man-made machinery.
Schrödinger is a name in physics to conjure with. I think even members of a quite broad general public would hear the name Schrödinger, associate it with Einstein. It goes with tough physics, it goes with quantum physics, and the fact that in 1944 Schrödinger published a book called What is Life? About biology and how biology would yield to scientific attention that physics could unpack biology, that was a light bulb moment for physicists.
I think it's a universal among all biologists that they've got physics envy. They really wish they were physicists, but they know they're not clever enough, and I know I'm not clever enough to be a physicist. As Lord Rutherford once said, there is only physics, everything else is stamp collecting.
In writing What is Life, Schrödinger helped shake off biology's stamp-collecting image and launched a period that would bring it to the forefront of science.
What I think was really useful about that book was the rigour of physical thinking of a physicist, which allowed the question to be properly framed. what he emphasized was the importance of thinking about the transmission of information from one generation to another. That was essentially the nature of heredity, and how you might think about coding information in molecules.
During the Second World War, Erwin Schrödinger was head of theoretical physics at Trinity College, Dublin. And it was here, in neutral Ireland, that he set about unleashing the rigour of physics on the messy business of life.
Schrödinger talks about life in physical terms, about life being an islands of order in a sea of disorder. And that's perhaps the best definition of life I can think of. And he also pointed out that it was a kind of twofold process. A living creature itself will die and return to disorder, but will have passed on its information in a molecule which at that time wasn't known. So there was a code involved too. So those two things, entropy and the code, both of which are physical ideas really, mathematical ideas, I think formed a lot of modern biology.
The same laws of physics and physical chemistry hold within the living body as outside. The simplest spontaneous bodily movement, say the lifting of my arm, would require the planned collaboration of billions of single atoms in their undetermined swerves if they should bring about the integrated action.
Science had started to make inroads into the secret of life in the previous century. Charles Darwin had proposed his theory of evolution, and Gregor Mendel had worked out the principle of how characteristics were passed from one generation to the next biologically through genes, though he had no idea what genes actually were or how they worked. Then things went wrong. The way that these ideas were misapplied is perhaps the clearest example ever of two rights making one colossal wrong, as demonstrated by leading British biologist Professor Julian Huxley.
What is the bearing of the laws of heredity upon human affairs? Eugenics provides the answer.
Eugenics was a 19th century idea that spawned a movement whose aim was to ensure that only the fittest survived. An attempt at biological engineering, breeding out those deemed to be a drain on society, people labelled at the time as defective.
Here is a man who, although normal, comes from a mentally defective family. Here is his wife, who is also normal. They have had 17 children. Let us examine the pedigree of these children. 5 of them died in infancy. 3 are still too young for an opinion to be formed of their mental state, a boy and 2 girls. Only 2 of the remaining children are normal, a man and a girl. The remaining 7 children are all mental defectives.
The people who set up the eugenics movement, who were studying human heredity, in retrospect knew nothing, and I mean nothing. Possibly less than nothing, because what they did know was simply wrong. But that didn't stop them from going into these dreadful practices.
In institutions such as this, all over the country, mental defectives are cared for. Once such children have been born, we must do the best we can for them. But it would have been better by far, for them and for the rest of the community, if they had never been born.
I think that before World War II, biology was somewhat of a backwater, not least because the interwar period is a period of frenetic development of military hardware. The scientific community, which was growing in stature, was regarded now as the great hope. It was what was going to defend Europe, defend the United States. The biological sciences were really, I think, on the back burner. The bright young men, and I use the term advisedly, went into physics and chemistry.
3, 2, 1, 0.
The irony was that while Schrödinger used physics to explain what it means to be alive, most of his bright young colleagues were using physics as a means of snuffing life out on an apocalyptic scale.
In the course of World War II, literally every able and eminent scientist was working for the war effort. And I think that out of that came a thoughtfulness about the meaning of life on the part of physicists, that they maybe thought more about philosophy. And I do think it characterizes the men of this generation. And coming out of the war into a time of promise and peace, wouldn't you then want to go where those thoughts had taken you? And that was biology.
One such scientist was Maurice Wilkins, an English physicist who'd spent much of the war at Los Alamos, developing the atomic bomb.
Certainly the atomic bomb business was a stimulus for getting out of physics into something else. And I think the immense destructive forces which were discovered in the atomic bomb were rather appalling. And so I felt that I wanted to work on things which were living and growing for a change.
Dr. Wilkins, what do you actually do? I mean, what do you actually do in your work?
The main thing in my kind of scientific work is to be able to fiddle with a thing, go on fiddling with it, and fiddle until everyone else has given up.
Wilkins settled into academic life at King's College London, where he began fiddling with crystallography in a bid to shed some light on the mechanism of genetics, on how what Schrödinger called a code might work.
Crystallography, the idea that you could shoot X-rays at a copper sulfate crystal or common salt crystals, and by looking at the way they bounced off, you could reconstitute what the structure of the atoms in the crystal were. That was pretty astounding stuff, and it made enormous advances in physics. And then somebody had the idea, Wilkins was one of the somebodies, of using that on the much messier system, which is biology. And to everybody's great surprise, it turned out that some molecules were really quite amenable to being treated in this way. And one of those molecules, which is very abundant in the body, is DNA. It was initially called the stupid molecule, because it seemed to be everywhere. It was in all cells, and yet it didn't seem to do anything.
Then, in 1944, a group in America performed an experiment which showed that DNA wasn't quite as stupid as everyone had thought.
They took strains of a pneumococcus bacterium, and the two strains differed. One was smooth, one was rough. And what they did is they extracted DNA from one of those strains, sprinkled it on the other, and transformed the character of that into the previous one, and therefore concluded that it was the DNA that conveyed that information. It wasn't protein, it wasn't lipid, it wasn't the other components, it was DNA.
DNA was the source of Schrödinger's code. It was where inheritance was written. So now the race was on to discover how it worked. It was already known that the DNA was made of simple sugars, some phosphate groups, and just 4 other chemical structures, the so-called bases of adenine, thymine, cytosine, and guanine. But how they all fitted together was a mystery. And the relationship between DNA and genes, the conceptual carriers of characteristics from one generation to the next, was another. These problems were what Maurice Wilkins was trying to solve with his X-rays. But Wilkins wasn't the only physicist in the game. In Cambridge, another out-of-work weapons designer was casting his eye over the same problem. His name was Francis Crick. But unlike Wilkins, Crick was not motivated by the horror of war. He just fancied a change.
After the war, I was, of course, in the Admiralty, but I really didn't want to go on being a scientific civil servant, as I was for the rest of my life. So I decided, well, what a marvellous opportunity. Here you are at the age of 30, you can go into what you like. But the problem was, what did I like? I noticed that I was telling some of my young naval officer friends things. I remember one about antibiotics. And it occurred to me one day, but you don't know anything about antibiotics. You're just gossiping about it. So I decided that the gossip test is a good one. That what you're really interested in is what you gossip about. So I looked what I was gossiping to people about in science, and it boiled down really to two regions. One was the borderline between the living and the non-living, and the other was the way the human brain worked. I knew nothing about either of the subjects. I decided it had better be molecular biology. It was nearer to what I knew. And so that's how I decided to work on molecular biology.
Well, I have to declare an interest, which is I knew Francis Crick from my childhood. He, as a young man during World War II, had been allocated as a very brilliant young physicist, had been allocated a key role in charge of a section that was developing deep-sea mines to blow up enemy shipping. And he, being the kind of man he was, a numbers man, decided that he didn't want to do mines that blew up all of shipping. He wanted to develop a mine that would only explode under minesweepers. And he did indeed do that. He did it successfully, and there are those who say that he helped win the war at sea because knocking out the minesweepers was exactly what was needed. Admiralty never came round to it. Admiralty thought of him as an uppity, uncontrollable, difficult young man who wouldn't take orders. He ran his section with great peremptory authority. So, here's this young man who's been given lots of authority, way ahead of what he would have got in a lab, and also has learned to challenge authority and get away with it, and indeed be able to say he was right. So he comes out of the war, and he's a very different kettle of fish from Wilkins. Amazingly links up, really fortuitously, with a madcap young man from America who went to university at 16 and has never towed the line. They form an intellectual couple. I don't want to say collaboration. They don't collaborate. They're a sort of, couple.
The madcap American was Jim Watson, a brilliant biochemist at the start of his career, who had recently received his PhD at the unlikely age of 22.
My first couple months in Cambridge were terribly chaotic when I went up. I went to the digs Max had helped get me on Park Place. Dismal place where the landlady wanted me to take off my shoes when I came in at night and didn't want me to flush the toilet after 10 in the evening. And after a rather short while, I wasn't very sympathetic and she threw me out. But I think the main thing was that none of these things bothered me because I'd met Francis.
Jim was certainly our first American visitor, and as soon as we met, we found that although we had very different backgrounds, we had a lot of things in common.
Neither of us were trained for what really interested us now. We both wanted to find the gene. We weren't organic chemists. We weren't anything else. We just wanted to do the best. We wanted to do the most sensible thing.
Francis Crick was a wild man, a man who encouraged wild behaviour around him. I was a student at Cambridge when I got to know the family well, and it was a big joke that if you stayed out overnight as an undergraduate in those ancient days, you had to stay with an MA of the university. So you could go to one of Crick's insanely wild parties and stay the night because he was an MA of the University of Cambridge. But I'm telling you, I shouldn't have been at those parties aged 18. And the thing about Jim Watson was that he was a prude and he was hopeless with girls. And he just tagged along with Francis. He just always wanted Francis to somehow get him into the thick of it, but he never did because he was sort of somehow too pusillanimous himself.
Crick and Watson's intellectual common ground was in the idea that they might be able to determine the structure of DNA. That challenge was something Jim Watson had first heard described by Maurice Wilkins at a conference he'd attended in Vienna.
Then I suddenly became aware there existed someone who actually was trying to solve the structure of DNA, which seemed to be the likely candidate for the gene. Well, but Maurice was serious, deadly serious. And I tried to talk to him, but Maurice, I figured, he's English and doesn't talk much to strangers. And so I left in sort of the vague feeling that it'd be nice if I could work with Maurice, but it wasn't a sort of obvious coming together of like minds.
Crick and Watson's approach to the problem was to try to imagine how all the known parts might fit together, build scale models, and talk about them.
Well, Francis likes to talk. It's his dominant quality. I think he doesn't stop unless he gets tired or he thinks the idea is no good. And so, since we hope to solve the structure sort of by talking our way through it, Francis was the ideal person to do it.
In London, Maurice Wilkins and the King's Group were actually doing something about the problem. Ray Gosling was a research student at King's at the time and helped with the X-ray crystallography experiments.
This is the original camera that we took the first DNA specimens on in this lab, and we had a fairly weak X-ray beam, so that we needed lots of material in the beam to get a diffraction pattern. And you see there a specimen made with 30 to 40 fibers of DNA wrapped around this little metal jig there, and we placed that in front of the X-ray set, and after about a day we had a diffraction pattern.
The data the experiments produced was found in these fuzzy photographs, pictures of how the X-rays were scattered by the DNA crystals. These, it was hoped, would provide the crucial evidence for DNA's elusive structure. But the results they were getting were disappointing. The pictures were too fuzzy. So because none of the group were expert in the technique, an expert was hired. In 1951, Rosalind Franklin arrived to help sharpen up the photos. Or so they thought.
She actually arrived while Wilkins was away, which is probably about the most unfortunate mishap in the whole story. Wilkins was away. He was expecting her, but she had come much later than she was supposed to come because her work took longer to finish in Paris than she'd expected. And she arrived in the laboratory. She decided what she wanted to do. She decided who she was going to work with, indeed who was going to be her graduate student.
When she came first to the lab, Wilkins was in America, and the interview took place in Professor Randall's office between Alex Stokes, myself meeting Rosalind for the first time. And I can remember my own feelings at that interview. It was very clear that as a research student, I was being formally passed from one to the other, and that not only was she being given the problem, she was being given an assistant to work with her on the problem.
Rosalind didn't see herself as collaborating with Maurice, but there was always this tension between the two.
I think Maurice, when he brought Rosalind in, sort of afterwards regretted he'd given away his problem. That is, he thought he needed help, brought in someone who was a trained crystallographer, and then discovered that it wasn't his problem anymore.
So it was a catastrophe. She didn't share and he didn't share, and I have no idea whether she was a sharing person or not, but certainly the conditions under which she was working meant that putting her arm over her work was absolutely, the starting point. She certainly wasn't going to let Wilkins have what she was doing.
But how did you get on with Rosalind?
Terrible.
You make out, Jim, you're sort of some male chauvinist pig, but I think the real thing it wasn't that, it was the fact that she didn't think you knew much crystallography.
To which she was totally correct.
Rosalind was rather prepared to discount them as being very serious competitors. I think there was a general impression in the scientific community at that time that they were like butterflies. They were flipping around with lots of brilliance, but not much solidity. And obviously, in retrospect, this was a sort of ghastly misjudgment.
It was a ghastly misjudgment because the Cambridge butterflies were actually getting somewhere by building their models, talking, and perhaps more importantly, listening.
In those days, there was a brilliant young theoretical chemist in Cambridge called John Griffith. He died quite recently. And he'd found that adenine stacked nicely on top of thymine and guanine on top of cytosine. He'd been thinking along those lines at the time, but I didn't know that. So I said to him, well, that's all right, that's perfect. That's all we need for a replication scheme. The relative amount of the four bases in DNA had been worked out by a nucleic acid chemist called Erwin Chargaff a few years before this time. And it happened he was passing through Cambridge, and he told us about his results, which were that the amount of adenine equaled the amount of thymine, and the amount of guanine equaled the amount of cytosine in all sorts of DNA, wherever he looked, although the other ratios were all over the place. Well, the effect on me was electric, because I saw immediately that this is what you'd expect from a scheme like John Griffiths'. So I was very excited. I didn't mention it to Shogef at the time, because it was work I was doing with John Griffiths, but when we checked it all out, we could see the two fitted together.
Crick and Watson now got word from the US that the legendary chemist Linus Pauling was also working on the DNA problem. Armed with a draft copy of Pauling's soon-to-be-published ideas, Watson went to King's to discuss them with Wilkins.
Morris read it and, in his usual way, didn't convey sort of enthusiasm one way or the other, but I guess sort of said yes, he didn't think Linus was going to get the right structure. At the same time, however, he sort of let loose the bombshell, at least to me, was that actually there were two types of DNA X-ray photographs. There was the form which I knew about called the A form, which gave this crystalline pattern, but there was a second form called the B form, and he opened a drawer and took out a photograph, and boy, I could hardly believe it. It was a perfect helix. It was a cross-like pattern, and he told me that it repeated at 34 angstroms, and that meant that it was a helix. I thought also, though, I should go in and see Rosalind. It was clear that she was sort of annoyed at my trying to tell her something about crystallography. And she came toward me. I thought she was going to hit me. So I quickly got out, at which point Morris was coming around, and she almost hit Morris.
Oh, God. Who hit who? I don't think anybody hit anybody. Actually, some people may have thought someone was going to hit somebody, but there certainly weren't very friendly feelings.
Watson escaped back to Cambridge in one piece, carrying with him the memory of the B-form photograph that Franklin had taken and Wilkins had shown him. Straight away, he started a new model.
So I cut some things out of cardboard and sort of made the right shapes and then pasted things on which would indicate hydrogen atom. Then I think I went off and played tennis. I would do it maybe 3 hours a day. I mean, it was hard to get at it in the morning, but because by the time you'd get in, there was morning coffee and then go for lunch, we'd have our walk, and then I'd come back and build the model. And sort of Francis was working on his thesis, and
I would look over my shoulder to try and see what Jim was doing.
I guess it's awfully hard to give up an idea of your own. So I started putting the phosphates in the center, maybe because it was sort of like a Pauling structure. Maybe if we wouldn't use ions, we could get somewhere. But Francis really wasn't comfortable with this and told me, why don't I try putting the phosphates on the outside? I can't really remember why you said that, Francis.
Well, it's because I think, Jim, that, you were obsessional about having them on the inside, and you produced a lot of phony arguments as to why basic groups from protamines had to go in. And in all collaboration, it's very important when one person has an idea that the other person criticizes it and, as it were, the devil's advocate. So just for the very reason that Jim was keen on having the phosphates on the inside, I thought he ought to try on the outside.
And suddenly I could put together A and T and G and C. Could hardly believe it. And Francis came in almost immediately and saw this.
Something came out of the model building that Jim had done, which he hadn't put in. And that's always the sign that you feel you're on the right lines. When something begins to click, which you hadn't actually put in your thinking of the subject, but you knew was there.
But even more important, Francis, by using these rules, A and T and G and C, we understood how the molecule replicated.
That's right. Everything from then on was clear. Everything was finished except the hard work. That's to say, producing an accurate model. I worked continuously for about 4 days, and then came the point where we saw that everything fitted, and I was so tired I went straight home and went to bed.
The structure of DNA had been found. A pair of sugar chains linked by the A, G, C, and T bases twisted into a double helix. But more than that, its structure made it immediately obvious how DNA could make copies of itself. If the strand of DNA is split apart, identical copies can be reassembled because each base can only pair up with its base partner.
There before us was the answer to one of the fundamental problems in biology: how do genes replicate? And it was very simple. and you couldn't miss it.
The great paper that was published the early 1950s with the structure of DNA is actually a masterpiece and beautifully understated, ending with what I'm sure must have been a Crick sentence about that the significance of this structure has not escaped us.
We used to occasionally Jim and I, just sit and look at the molecule and think how beautiful it was. And I remember an occasion when Jim gave a talk to a little biophysics club we had. It's true they gave him 1 or 2 drinks before dinner. It was rather a short talk because all he could say at the end was, well, you see, he's so pretty. He's so pretty.
The twin successes of the discovery of the structure of DNA and a mechanism for its replication was a critical moment in science.
I was a student in Edinburgh, and rather cleverly, my tutor told me to go to the library and to find the most important paper in biology. And I thought, this man is mad. How am I going to do this? And I walked in. These are the days when everything was bound into big volumes. And I walked past the journal Nature, bound in green, covered in dust, until you got to the 1953 issue. And it was battered, it was torn, the back of the cover was come off. You opened it up and 2 pages fell out, which were black with the grease of many scientists' fingers. And that was the Watson and Crick paper.
The paper may have rocked the scientific world, but so far as the public were concerned, 1953 was a year remembered for bad weather,
the waters rose with the wind,
a coronation, and the conquering of Mount Everest.
Edmund Hillary, beekeeper from New Zealand. Tenzing Norgay, Sherpa from Nepal, conquerors of Everest, May the 29th, 1953.
The structure of DNA doesn't even get a mention.
Biology really wasn't on the agenda, certainly not the public agenda or the press agenda. That was not what they were going to get excited about. The public was still in love with physics and chemistry, and they were. So if you look at the South Ken exhibition for the Festival of Britain in 1951, it's all physics and chemistry, all the excitement of molecules and the excitement, and then the excitement of radar and the excitement, not bombs, of course, nothing about bombs.
But biology, toughened up by physics, was on the march. New vaccines and medicines had whetted the public's appetite. And in 1962, when Crick and Watson, together with Maurice Wilkins, were awarded the Nobel Prize for Medicine, the double helix, DNA, had well and truly arrived. The BBC even commissioned this special programme about the prize winners.
This is to be a personal programme about these men. In a few moments, through television, you will be able to meet them and judge for yourselves what manner of men they are.
Biology was now at the forefront of science. The possibilities seemed endless.
I think one can reasonably predict that within the next 20, 30, 50 years, there will be an immense increase of knowledge about the higher nervous system and about ourselves. And I think myself, as a personal belief, it will radically change the way we think about ourselves as persons and also eventually as people in society.
The old rivalries were put aside, at least for a while. Though since the Nobel Prize was awarded, the public memory has been of Crick and Watson, and not of the others who helped them over the line.
But of course the Watson-Crick breakthrough was as I've said many times, a little sort of pinnacle built on an immense wide basis of chemistry, biochemistry, genetics, and so on. Essential work which people like Todd and Chargaff and many others had to work through before one was able to put that little thing, the three-dimensional structure of DNA, on top.
Perhaps least celebrated of all was Rosalind Franklin, who had provided the crucial evidence in the form of the B-form diffraction pattern that provided the final part of the jigsaw. Franklin had developed ovarian cancer and died in 1958, and so never knew about the prize or the public excitement around DNA. It's often said she was unjustly overlooked, but the Nobel Prize is not awarded posthumously, so the truth is less clear.
I sometimes wonder what would have happened had Rosalind Franklin been alive when that Nobel Prize was given. I don't really know what the outcome of that would have been. The Nobel Prize is only given to 3 individuals. Crick and Watson would have been clear. I suspect that Franklin might have trumped Wilkins. I don't know that for sure, but that, I think, would have been a definite possibility.
Though many contributed to the discovery, it was ultimately Crick and Watson who made it, and they had their own views as to why.
We weren't at least afraid of being very candid to each other, to the point of being rude. And if you don't have constant interchange and chatting together and saying what you think of the other people's ideas to their face, I don't think you can solve problems of this kind. We sort of pooled the way we looked at things. We didn't leave it that Jim did the biology and I did the physics. We both did it together and switched roles and criticized each other, and this gave us a great advantage over the other people who were trying to solve it.
See, what I think is interesting is that Crick and Watson do insist on how collaborative their own work was between the two of them, but I don't kind of regard two as much of a collaboration. I think it was more of an intense relationship, scientific relationship, and the fact that they absolutely knew they needed one another. They each needed the expertise from the other's discipline. They were absolutely not collaborative scientists outside, not in the least. For them, it was a game of who you could beat. In my hearing, I heard them laughing about Linus Pauling and not showing something to Linus Pauling because otherwise his lab might get there first. It was all about who'd get there first. That's not collaborative science.
Maurice Wilkins seems to have been all too willing to collaborate. He was, perhaps, a naive idealist and something of a victim of his understated demeanour.
Wilkins was an active communist in his youth. He was a spunky man. We have no knowledge of that, partly because, presumably, such activities as he was engaged in, he didn't exactly trumpet abroad. The only reason I know that he was that kind of man is because his MI5 file was released in 2010, and it is clear from there that he was regarded as a threat. Wilkins must have been a much gutsier man in his youth than the bits of film that we see of him would lead one to believe.
You interested in music?
Well, I used to be, but I'm not so interested now.
The reason we think of Maurice Wilkins as this irritable and actually irritating man, when my view is that as a young man he was probably quite dashing and exciting and communist and non-aligned, is because of his behaviour around the fact that Rosalind Franklin just wouldn't play ball, wouldn't be a member of a collaborative lab, and I'm sure in Paris had not been expected to be. She didn't get it, she didn't sign up for it, she hadn't signed up for it. The fact she wasn't allowed in the common room at King's London because women weren't allowed in it really didn't help. And therefore Wilkins' collaborative endeavour embraced everybody outside, but broke down in his own lab.
And looking back on it, of course, it's quite clear that if you regard sort of getting the structure of DNA as a race, that we'd lost the race very early on because we didn't find it possible to work together.
Regardless of who won or lost, learning the structure of DNA and finding out how it might replicate were huge Nobel Prize-winning discoveries. But knowing these things revealed nothing about how DNA actually turns mindless chemicals into trees, frogs, parrots, and people. How our genes, now realized to be discrete sections of DNA, give us unique anatomies and personalities, making each of us who we are. This became known as the coding problem.
So the next important problem was the coding problem. Of great interest to Crick. By this time, Watson had left, gone back to the United States, and Sidney Brenner turned up.
Sidney Brenner was a reluctant doctor from South Africa who'd hung up his stethoscope in favor of research as soon as he possibly could.
Well, I grew up in a small town in South Africa where my father was a shoemaker. At the age of 15, I went to university. And I was strongly advised that for the subject I was interested in, which people thought was biological chemistry or whatever. The only jobs existed in medical schools. And so I was told, you've got to get a medical degree because no one will employ you in a medical school without a medical degree, which I did. It was another 4 years. It was actually 4.5 years because I failed my finals in medicine by diagnosing one lady as having an acute attack of the use of McLean's toothpaste rather than something else. I was right. She actually had used McLean's toothpaste. I was told to smell her breath, and that's all I could smell. However, but I did return and did the other 6 months and finally passed. It is a very difficult experience doing science, I think, in a provincial atmosphere. All this changed for me when I came to England to work for a PhD.
If I laid out all those great scientists who I had the privilege of knowing as a child, Sidney Brenner was the funniest one. Sidney Brenner was just damn funny and comfortable to be around. He was a sort of very gregarious, very open-faced, open everything man. He rode a huge motorbike, you know, but he was very sort of stocky and together, and I thought he was quite glamorous.
Now, Sidney Brenner, another one of these brilliant characters, who's also extremely witty and humorous. I perhaps know Sidney the best of all the characters, perhaps together with Jim Watson, and I always seek him out if there's a dinner so I can sit next to him, because I will be highly entertained throughout dinner. Just like humour, you put different things together, you juxtapose different sorts of ideas and thinking, and they're funny. It's the way he also carried out his science. He would put different things together and see how things could emerge from it. I think that they were related. The creativity of humour, I see also was in his creativity of tackling the coding problem.
Now, the coding problem was enormously attractive because, in a sense, it looked possible that you could crack it, solve it, without doing any work at all. There would be something in the sequence that you could then sit down and write down the code.
Once again, we have a collaboration. This time, it's Crick with Brenner. Watson's gone back to the United States. During the period that Brenner and Crick were working together, there was one of the greatest experiments of all time to determine what's called the triplet nature of the genetic code. Because the problem was this. In DNA, you have a language made up of 4 letters, and in proteins, the language is made up of 20 letters. So you can't encode for 20 different letters by 4 alone. They have to be read either in 2s or 3s or 4s. And the key experiment was what the number was. And there was an absolutely genius experiment done where Crick explains this, that he introduced mutations, and if you introduce 1 mutation, then you would destroy function. If you introduce 2, you would destroy function. If you introduce 3, you got the function back again, because all you did was lose 1 amino acid, and then the rest was okay. And this is a brilliant piece of abstract reasoning which came to a concrete conclusion.
We had discovered that the dye proflavin does cause additions and deletions of bases in DNA. DNA consists of a one-dimensional sequence of 4 different building blocks, the bases. The proteins consist of another language, the amino acids, of which there are 20. You need more than 1 nucleotide to correspond to an amino acid, because there are only 4 of the first and 20 of the latter. Indeed, you need at least 3. So we suspected the code was a triplet one. We also suspected that it was read from one end 3 at a time. Imagine a gene being composed of the triplet CAT, C-A-T. So we have cat, 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 cat. you can see that if you delete one of these nucleotides, beyond the deletion, it's converted into gibberish. ATC, ATC, and so on. If you delete yet another nucleotide, you will see you get the second form of gibberish, TCA, TCA. But if you delete a third nucleotide, you convert it back to the original message, cat, cat, cat. It absolutely worked. It proved that the code was non-overlapping and a triplet one, and then, as a joke, we later called the triplet a codon, the unit corresponding to one amino acid.
So this was an example of the sort of thinking, the brilliance of the thinking, that was tackling problems in rather simple ways, not fantastically sophisticated equipment and so on, but brilliant logical reasoning.
What had been accomplished as a result of Crick and Brenner's discovery had opened the door to the ultimate possibility, that DNA was the book of life, that the units of inheritance, the genes, were simply specific sequences of A, C, G, and T held in the DNA, and that it could all be read in its entirety. But before the final assault on the stuff of life could begin, another problem needed solving. Even though the structure of DNA was known, and it had been shown that DNA coded for proteins, actually reading the exact order of the bases was proving impossible. Enter Fred Sanger, who, like Crick and Brenner, was from the molecular biology lab in Cambridge.
Fred Sanger, who I also know a little bit, is a very humble man, very somebody you hardly would notice. You know, you'd think he was the gardener, I'm sure, in Cambridge, rather than, you know, one of the greatest scientists of his time.
Fred Sanger had already received a Nobel Prize for discovering the structure of proteins. Now he turned his attention to how to read or sequence DNA's elusive code. For Sanger, there was a useful similarity between proteins and DNA.
One became much more interested in nucleic acids. Again, it was a long-chain molecule similar to the proteins, so that there was a similar problem there. And I thought maybe my abilities would be useful in that field, and we might be able to do something in sequencing them, because clearly the sequence was the important thing about them.
I think Fred Sanger, who really pioneered the notion of sequencing DNA, using what in retrospect was a clumsy method, but was the only one that was then available, is again a rather forgotten figure by the general public. And he set out to do something which certainly when I was a student of genetics, and when I was doing my PhD in genetics, and even when I started my career in genetics, it seemed fundamentally impossible. The idea that you could read off a complete genome just seemed completely out of the window, let alone that you could read off the human genome. But Sanger started the job with this method of labeling the bases one at a time and chipping them off and separating them and just reading them ploddingly slowly from one end to another. And he played a central part in the technical change which turned genetics into the modern day equivalent of anatomy.
Fred Sanger's work earned him another Nobel Prize and opened the door to a tantalizing prospect, that the DNA of an entire species, its genome, could be read, that its genetic code could be solved. John Sulston is the archetypal left-leaning, Guardian-reading, muesli-eating scientist.
I like muesli, you know. I'm not doing it for any particular reason, I suppose. That's what I mean.
Sulston is a man with a very clear idea of what is important in life and what is not.
I suppose simple. Rampant anti-consumer. Well, anti is wrong. No, non-consumerist is the point. But on the whole, the presumption is that one is not going to buy things rather than that you are, if you see what I mean. You know, what do you want, really? I mean, It's thinking and reading and talking and all of that that matters, not actually having stuff, unless the stuff's there for a purpose.
His low-key domestic arrangements give few clues to his standing in the academic world. Like Sanger, Wilkins, Watson and Crick, John Sulston has a Nobel Prize. It was awarded for cataloguing the development of every single cell in the nematode worm. Years of research, culminating in 18 months of intensive observation. 8 hours every day, looking at worm cells through his microscope.
I thought it was rather fun, really, you know, it's a job of work. You don't have to worry about what you're going to do the next day. You know what you're going to do. You're going to go and watch some more cells divide.
What he did next was even more tedious. To work out exactly why the cells behaved as they did, Sulston needed to understand their genes. And to do that meant sequencing them. All of them.
Before we began, people said, oh, it's not worth doing that. What you ought to be doing is to study real biological problems. Look, John, they would say, you've done all this cell lineaging and you ought to be getting mutants. You ought to be finding out how the cell lineage works. And I said, I'm not sure. I think it's more complicated than that. We really need to get at all the genes. And so I started this business of looking directly at the genome as a whole.
The nematode worm genome was the proof of concept. Now the way was clear for the ultimate prize. The mild-mannered worm scientist found himself running the British part of a global effort to read the human genome. From its outset, Sulston believed that the Human Genome Project was a noble undertaking for the benefit of everyone.
All the labs met in Bermuda, and we met in Bermuda precisely because it's a rock in the middle of the Atlantic, so it's a sort of neutral place. It was in the off-season, I should add. So we were there, and I remember standing at the board there, trying to construct with the assembled labs a statement to the effect that all of us would release all of our data all the time, and we would not try to take patents. And we came out with what have been called the Bermuda Principles, and have actually informed quite a lot of endeavour since.
It was all done in a very gentlemanly and gentlewomanly way, and everybody was very nice and decent and, let's say, rather British about it. And then suddenly into this calm Cambridge environment came a bombshell, this chap called Craig Venter.
Craig Venter had been the team leader of one of the genome groups in the US, but had decided to go it alone. There was, he decided, money to be made in the human genome.
Get away! There were lots of biotech companies that viewed genes as commodities. People started assuming you got a patent on a human gene, you'd get $1 billion drug.
This went against everything Sulston believed.
Human genes should be owned by the public, not owned by any particular person or even corporation or even government. They're publicly owned.
John Sulston now found himself as the reluctant defender of the people's genome. He was now in a race. Every gene that he identified and published was one that couldn't be patented by Venter. He secured extra funding to ensure his UK group could complete a third of the genome, and jetted to America to stiffen the resolve of the National Institute of Health, who were on the brink of cutting a deal with their former employee's new company.
I think it did turn things around because NIH were in fact making moves to compromise it because they thought they were dead. They thought that, Congress would just say, well, right, now you've got a company doing it, you're not allowed to compete. But by us coming in and saying, well, the Brits are going to do a third publicly, it really stiffened the backs. You can see the sort of moral force it had.
Not for the first time in the story of DNA, things got nasty.
John wanted to sequence the human genome. I wanted to sequence the human genome. I can't fault him for wanting to do that. But it was more important to him to be the one to do it than to enable it to get done quickly.
I suppose I am anti-capitalist to the point where I feel the companies are completely unnecessary. Yes, that's right. I think we'd get on very well if we did all of that discovery in universities, frankly.
They gave him the Nobel Prize for counting cells in a worm, right?
The absolutely debilitating diseases, AIDS, of Africa and other developing countries, they cannot be dealt with on a capitalist basis because there's no market, there's no money.
So you now have two sort of programs going in parallel. One, a public program set up for the public good. A second, a private program, ultimately leading to the public good, but in the meantime going to make a lot of money.
Our efforts are at least equally important, if not more so, because it forced them to get going and get off their butts and actually do something.
Ah, yes, Celera lit a fire under the human genome. They always like that image. But I think objectively the facts speak against it. You know, I really don't think that it was helpful. I think we'd have finished pretty much next year anyway, and without all the fuss.
Salston's a much, much more traditional scientist, much more of the Maurice Wilkins type, the sharing, collaborative, making the world a better place. I wouldn't have thought it would have crossed his mind that he could make his fortune. I think Venter saw very early on he could make his fortune. I don't think that's a bad thing, but he saw it.
Well, Venter was simply, you know, he was... How can I summarise it? Venter was an American. Salston was British. That, I think, summarises the entire problem.
But ultimately, it was politicians, British and American, who came down on the side of the people's genome.
New genome science and its benefits will be directed toward making life better for all citizens of the world, never just a privileged few.
In 2000, Bill Clinton and Tony Blair announced that the race was effectively over and that no one would be making any money by patenting genes.
When the public project guys, i.e. you and everybody else, got Tony Blair and Bill Clinton to make that announcement early last year when they said that they would like to see unencumbered access to this data, it wiped a kind of $150 off his company's, Venter's company's share price, didn't it?
It's a natural thing that would drop.
But it must have made you smile. Put a smile on your face. Well, it has now. Anyway. Thinking back about it.
Yes, but the trouble is, I'm at the same time. It's true, I smile, but I mean, these are minor victories or skirmishes in the whole thing.
Much has been made of the importance of the reading of the human genome, of the medical benefits that will emerge from its now cracked code. That maybe it even holds the secret of life itself.
We cannot understand what life is without understanding the genome. But it is only one part of the problem. And we need also to understand how living organisms work, how cells work, how tissues and organs work. But without the human genome sequence, we can hardly address the problem. It's crucial, but it is not the only thing we need to know.
Since the middle of the last century, the murky business of inheritance has been dragged out of an age of near witchcraft to one where individual groups of atoms in what was once considered the stupid molecule are now recognized as agents of heredity. Crick and Watson's identification of DNA structure, informed by the crystallography done by the King's Group, Sidney Brenner's solution to the coding problem, Fred Sanger's work on sequencing, and the global effort to read the human genome has revealed what some might call the code of life. But in reality, DNA, coding, and genomics is only the start of the next chapter.
Stories from the Dark Earth sees science challenge what we thought we already knew. Archaeology coming up on BBC4 next.